Hello, everyone. Coming at you from a little bit of a different location in my backyard. So today we're going to talk about vaccines and specifically vaccines against coronaviruses. Um, by now you should have finished the first page. This first one page from your search for speeding up vaccines against new coronaviruses. That's the first line that says on the outside. I posted the link on Google Classroom yesterday. So the answers were all in that article, but just to discuss the questions anyways. We kind of talked about this last time, but vaccines, what are they made up of and how do they work? So now that we know how coronaviruses infect cells, let's talk about how vaccines work. So they're made up of those weakened or killed parts of viruses. It can either be a whole virus and just a killed version of it, or it can just be a weakened, alive, but weakened form of it, or you can just take the outer part of the virus, which is what your immune system actually recognizes as the outer part of the virus, and then they can use that for vaccinations too. But your immune system then is activated and starts making antibodies specific to that virus, specific to the outside of that virus. So any protein that's on the outside of that virus, like the coronavirus has those little spiky proteins, that's what that's what your immune cells will recognize and start making antibodies against. So, and remember what the antibodies do is they actually capture the virus. So those little V things that we saw in the last episode, where they actually captured it on all sides. That's what the antibodies do, and they help your other white blood cells attack and recognize those viruses and all other infected cells of that virus. So they do a lot of different things, and they do it very quickly if you're already vaccinated. That's the key point. They do it very, very quickly if you already recognize that virus, which is why usually when you're sick with it once, you can't really get sick from it again because your body is quick to react to that. So they have these new forms of vaccines. One is the Inovio scientists, that company that made the MERS vaccine, and theirs is a pretty standard vaccine with a little novel twist to it. So remember that your coronaviruses are made up of RNA. So what they did was they switched that RNA back into DNA. And then once they have that DNA, they can make that into proteins. And so that's what they did. They actually put the DNA into a bacteria. And we use bacteria to make a lot of proteins for us, like insulin. So they did the same thing. They took that DNA sequence and then put that through bacterial cell, and it produced the proteins that were on the outside of the virus. So they made the spike proteins and all the proteins on the outside of the coronavirus. So you get the parts that your immune system recognizes without the parts that make you sick. So it's kind of a bonus. And the good thing is that those bacteria can make thousands of copies of those proteins very quickly because they reproduce so quickly and they make proteins so quickly. So they're a good thing to use to make proteins, like vaccine parts. And that was their process. And then they would use these proteins to make the form of their vaccine. They would just use it as a traditional vaccine. However, that those tests are still far away. They don't know how effective it'll be. They haven't done safety tests yet. It hasn't gone through the FDA. There's still a lot of steps to take there. So we're still about a year away from a vaccine at minimum. We have Moderna's mRNA vaccines, and this is a novel, a completely new approach to vaccines that I honestly had to learn about as well. So they're stimulating body cells to actually make the vaccine components. Okay. So what they're doing is they're making your own cells make the viral proteins and stimulate the immune system. So the goal is that the mRNA that they inject into your cells will give your cells instructions to make those viral proteins. And then your cells would actually be the ones to make the proteins for the vaccine. Just the outer part of the proteins, the outer part of the virus. You would not be making the genetic part of the virus or any of the parts that actually make you sick. So it's just taking the stuff that starts activating the antibodies, making your cells produce it, and then your cells release it, which starts activating those white blood cells to start making antibodies. So your cells would be doing the work with the mRNA kind of blueprints, instructions from the injection you would get. And this would stimulate your immune system without making you sick. Then you have that molecular clamp, which is another group. They called it the molecular clamp. And so what these things would do is they would make proteins. Your cells would make a protein that would attach to the spike protein and basically further initiate any vaccine that you would get. It would put that spike protein into just the perfect shape to initiate your body into making antibodies. So it would just kind of lock it into the perfect position for your white blood cells to recognize it and start churning out antibodies. So that's another thing that they're looking at too, is that molecular clamp, just that protein that's going to attach that the virus right in that right position so your cells can recognize it, which means you'd mount a very quick immune response. Why can't we expect it soon? Well, kind of as we've talked about a little bit, you have to do tests of effectiveness of the vaccine. So first of all, it has to work, obviously. You don't want to inject yourself with anything that doesn't work. Then you have to do safety tests. So it also can't make you sick with anything else. Often these safety tests will take a year by themselves because they have to do them in animals. Then they have to do them in humans. And they don't want to rush anything into production that's going to make someone sick long term. So that's why it takes so long to do those safety tests, is they want to make sure that they're not making people sick after a while. And then you also need a company to make a really a vast amount of these. Because think of how many of these vaccines we would need globally. Tons. We need billions. So you need a company or more than one company to be able to work with them to actually develop the vaccine. And that can take time as well. So none of these things are quick. You also have to grow up the virus and actually make the stuff to begin with, to do your tests. All of those things take time. It takes about a week just to grow a plate of cells. That's what I used to do in the lab, is you'd have this flask that's about that big, it's kind of a square, and you'd put a few cells in there and they would attach to the bottom. And in order for those cells to grow and be all the way across the bottom and start producing stuff for you, that takes about a week by itself. So you can see how much time these things will take, and why it's not going to be ready. It's not going to be ready as soon as someone starts developing it. Even if they have an idea quickly, that doesn't mean it's going to take place quickly. And then you have alternative vaccines. So they talked about just putting antibodies into people, just skip the middleman of the vaccine and just inject the antibodies right into somebody. And there's more than one way they've talked about doing this. And one is something that they've done before, which is take blood serum. It's basically the part of your blood, like the liquidy part, that's not any of your cells. So you take the red blood cells and the white blood cells out. It's just the liquidy part of the blood. And you put that in somebody, and that has your antibodies in it. So someone who recovered from the virus would have antibodies against that virus. And so theoretically and non-theoretically, because they've done this in people, you can take that blood serum and inject it into healthy people to prevent them from getting sick. And you can also treat people who are in the early stages of infection by actually giving them part of the blood serum as well. And that would give them an antibodies to start fighting the infection right away. Then you have mice that could also grow vast amounts of antibodies that you could inject in somebody, because the blood serum from another person doesn't always work. It's not always very effective. It works sometimes, but that's not really a reliable thing when you're talking about a treatment. So they've also talked about having mice just produce the protein, produce the antibodies, because mice make these antibodies as well. And if you give them instructions to do so, then they make the same antibodies we do. So then you can take, grow them up in mice, kind of use the mice to produce them, use the mouse cells to produce them. And then we can take those antibodies and use them to treat people and prevent infection. So those are just a couple of the treatments that are being looked at. But even with the blood serum test, you're still talking about safety tests. You're still talking about FDA submissions. It has to be produced by in high billions of amounts. Because pretty much everyone has to receive one of these things, everyone in the world, unless they've recovered from the virus already, which honestly is not that many of our whole population quite yet. So that is our vaccine talk about coronaviruses. So now we're going to talk about like why vaccines, why do they work? Why is this necessary? Why do we even make vaccines? And we're going to start investigating this term called herd immunity. Herd immunity, like a herd of deer and immunity. And that's what we're going to be talking about next. So your assignment is going to be, and there'll be a video clip attached to this that is also linked in here, but I will post it on the post. But you're going to do this page, which is the following page from what we just did, and then just the next half page, and that's it. So that's number 1 through 4. You will need these R0 numbers, okay? I think you can see these. So R0, which we will talk about. I'm also going to have a video so you can watch R0. R0 is, just so you know quickly, basically if I'm infected, it's the amount of people, if I'm infected with coronavirus, how many people I can infect on average. So just taking on average, the average person with coronavirus, how many people that one person infects on average for the whole population. So you have 3 scenarios we're going to investigate. One in which scenario A is 2.2. So an average person with coronavirus will infect 2.2 people. Scenario B would be 1.88. Scenario C would be 1.43. And the lower the number of people that a person who's infected transmits that to. So scenario A you can think of as like, scenario A is like the worst possibility. Scenario B is like a medium possibility. And scenario C is like a minimum possibility. So if we take all the precautions, you can think of it like that. All right, we'll see you tomorrow. Bye.